Bon, mais on va commencer tranquillement. Bonjour à tous et à toutes. Bienvenue aux conférences du CRCHUM. On a le plaisir aujourd'hui d'avoir avec nous Ben Creelan du Moffit Cancer Institute qui va présenter sur l'immunothérapie à l'aide de TIL. Welcome everyone. We're really glad today to have you attending the CRCHUM conference, scientific conference. We're really pleased to have Dr. Ben Creelan with us today. I see many names here from the institution and outside the CRCHUM as well. Dr. Creelan, you will be talking to students as well as attendings and scientists interested in this field. So Dr. Creelan earned his MD from Jefferson Medical College. He completed an internal medicine residency at Thomas Jefferson University in the Department of Internal Medicine. He then completed the Hematology Oncology Fellowship at the University of South Florida at Moffit Cancer Institute, and a master's degree in clinical and translational research through the NIH K30 sponsored SPORE program. Dr. Creelan is really a leading expert in the field of lung cancer in general and lung cancer immunotherapy and personalized treatment in particular. And without further ado, Dr. Creelan, it's really a pleasure to have you with us today.
Thank you so much. I'm really honored to be able to present on a field which is near and dear to my heart, which is the cell therapy, live cell therapy for lung cancers, something which we never thought we'd be talking about 10, 15 years ago. And so, again, I'll be trying to break things down into concepts so that it's usable for everyone and make sure that the fundamentals are covered because, again, I think this is an area which is becoming more important over time. So these are my disclosures, and I do some consulting or advising for several cell therapy companies and have received research funding to my institution for non-monetary services like drugs or conducting clinical trials. So I'm going to start out with an overview of cell therapy. And then I'm going to update you on the most prominent or promising recent data across this field. You know, overall, there's three main forms of adoptive cell therapy, right? You can either take genetically manipulated T cells, like CARs, TCRs, where they're recombinantly changed, or you can have non-gene modified approaches, where peripheral blood cells or tumor infiltrating lymphocytes are removed from the tumor, selected, and then cultured, right? So we can do this with T cells, we can do this with macrophages, we can do this with all sorts of immune cells. But today, I'm going to be focusing on T cells. And now gene modification is particular to TCRs and CAR T cells, but TIL can also be gene modified. And we have several trials coming up which do have gene modified TIL. So how do I think about this? When we're evaluating a cell therapy, I think the key thing for all the students on the line, for all the folks on the line, especially students, think about the antigenic target of the cell therapy before you start thinking about other things. Try not to be distracted by the technology, by the engineering, by the editing. Biotech world will always try to dazzle me with fancy words, but the key to understanding any therapy is always its antigenic target. And this also applies to bispecific T cell engagers or BiTEs. And you know, if you're trying to decide whether to open a trial, the key thing is if the target is a bust, then the drug will be a bust. So, you know, think about what your target is. TCRs usually target retained genes from development, as you can see here, like ontogenic proteins, MAGE, cancer testis antigens, or more commonly now, public neoantigens like KRAS G12V or EGFR. So we have this spectrum of scalability and personalization across the cell therapy. Whereas if we get more and more personalized, we also get treatments which are less and less practical to implement. And you'll see this theme throughout the presentation. So try to take away this theme, again, that you can have less personalized treatments, which may have, again, target more normal cells, but they're more scalable. And the more personalized cell therapy starts to become more unwieldy. And then we have unsorted T cells, which kind of run the gamut here because we don't know when we make an unsorted TIL or unsorted T cell that what the actual target is. So how does this work? Well, tumors are full of aberrant antigens, which our T cell repertoire recognizes as foreign, the same as COVID-19, right? And our antigen presenting cells obviously present these through MHC class 1 or class 2. So most of these T cells are targeting random passenger mutations incurred from the DNA damage during cancer development. Very few are shared or public neoantigens. And in fact, experimental evidence we know from National Cancer Institute shows that only 1 in 70 of these are strongly antigenic. So only 1 in 70 cancer mutations actually yield an effective antigen. And so with adoptive cell therapy, what we do is we remove those immune cells and we then pre-treat the patient with lymphodepletion chemotherapy to make space for the homeostatic expansion of the infused T cells. This is essential, and it's called lymphodepletion chemotherapy. With conventional T cells, this is believed to be essential because if we just give the T cells, your body gets rid of them. And again, since the ex vivo cultured T cells like TIL or TCRs are cultured in IL-2 with no additional activation domains or things to make them stimulate, we actually have to give additional exogenous IL-2 afterwards to promote their engraftment in the body, right? So the preparative regimen is very important, and we infuse this army of billions of T cells, but then we also give exogenous IL-2 afterwards in the conventional approach to support the T cells engrafting in the body. So a lot of folks have tried to get around this, but there's been trends in adoptive cell therapy over the past decade that taking shortcuts with lymphodepletion tends to result in lower response rates, whether we're talking about CD19 CARs, chimeric antigen receptor T cells, and lymphoma, whether we're talking about TCRs targeting cancer testis antigen, which was shown in a poster last year in sarcoma, or TIL at the National Cancer Institute. The more lymphodepletion that was given, generally the better the responses, the better the homeostatic expansion, the clonal expansion of those cells once they go into the body. Same thing with cell dose. The higher the cell dose, whether it was TIL, whether it's neoantigen-specific T cells, or whether it's TCRs, again, this all kinds of adoptive cell therapy, the higher the cell dose, the higher the response rates in general. Of course, some of you would say, well, there's no randomized clinical trial to show that, but this is just a trend, again, as you look through the literature. Same thing, albeit weaker, with IL-2. The higher the dose IL-2, the more IL-2 as part of that regimen, there does tend to be a trend towards better responses. Although there's no direct comparisons here, and certainly some patients have responded with just minimal doses of IL-2. So that brings me into an overview of one of the cell therapies that's near and dear to my heart, which is TIL. And TIL, as we know, has been around for decades. It's already enriched in tumor-specific T cells because we're taking it straight out of the tumor, right? It's polyclonal, so these T cells could recognize anything in the tumor whatsoever. It's totally unbiased from that standpoint. And of course, there's no HLA restriction because it's coming right out of the patient, and there's no gene modification required per se. We've never really seen on-target off-tumor adverse events with TIL, with the exception of things like vitiligo, which is was seen in melanoma TIL. But there are problems here, right? You have stochastic drift of those T cells in culture, where you may end up with a lot of irrelevant bystander, non-tumor specific T cells at the end of this long, several week-long culture process. And you also have the problems of the expense and the encumbrance of the surgical biopsy. Despite that, the hallmark of TIL is this potential for durable T responses, which can last for decades. And that's partially because of memory T cells. Normally, normal T cells last for what, like 7 days in your body, and then they're gone. And then there's new ones made. Memory T cells can last 60 years in your body. So this probably is the reason why we can see this ability to eliminate the very last melanoma cell, or at least patrol, continuously survey the involved organ to actually prevent recurrences in a really durable manner. Again, patients that were treated in the late 80s, still in remission with metastatic melanoma. And here's showing more of data about this. Again, if you can get a complete response, your chances of having a durable remission are extremely high, and you may very well be cured with metastatic disease. Now, what about most of this data? You're going to tell me, while it's impressive, this historical experience was mostly an anti-PD-1 naive melanoma, right? So TIL is also active in post-PD-1 progressed melanoma, but the response rate's less. And though the responses are durable, it seems like the duration of prior PD-1 exposure does correlate with the odds of TIL response. And I think this is the key question: is in the long run, is it better to be giving this type of therapy up front for all patients, or wait until they progress on immune checkpoint inhibitors? Because we know that also the survival looks a little worse in the PD-1 experienced melanomas. And I think that by resecting a tumor before it's seen an immune checkpoint inhibitor, we're going to see, we do see better durability and memory of those infused T cells. In contrast, this basically you're a horse jockey that's been whipping this horse around a track, and now we're trying to culture T cells from that horse. Well, the horse is exhausted. It's already kind of run its race, and those are not the best T cells to be infusing the patient. So again, across trials, we're seeing better responses in PD-1 naive patients, or at least tumors that have been resected from PD-1 naive patients. Now, the National Cancer Institute has shown that most patients do have TIL, tumor-infiltrating T cells, capable of neoantigen recognition, true across a variety of refractory epithelial cancers. So this is not unique to melanoma. Everyone's tumor, in theory, has T cells within it, which recognize those cancer mutations. They may have very, very few, but there should, in theory, be T cells there. So now I'm going to go into our lung cancer experience. This historical record led us to say, hey, what if we did this in lung cancer? And we did. We did a phase 1 trial in patients with stage 4 lung cancer who had not received prior PD-1 therapy. We resected tumor, usually through a metastatic cervical lymph node or a VATS thoracotomy to collect a metastatic pleural nodule. And then after resection, patients had nivolumab for 4 doses and 2 serial CT scans. Those that were responding to nivolumab just stayed on it, and we kept the TIL product frozen, cryopreserved. But the patients who were not having benefit to nivolumab went ahead and received lymphodepletion Cy/Flu and the TIL at standard doses, and it's intermediate infusional IL-2, so kind of an intermediate dose of IL-2. And a month later, we got a CT scan and did an apheresis to get antigen-presenting cells to do functional testing. So the majority of these patients who were actually progressing, enlarging, worsening on the nivolumab had a decrease in their tumor size. In fact, we had 1 confirmed partial response, 2 confirmed complete responses, and 3 unconfirmed partial responses of 13 patients evaluable. So a really surprising thing to take away from it was that, this trial included 50% never smokers. And surprisingly, several patients had durable remissions, including complete responses, which are ongoing to this day, with no correlation between smoking history, PD-L1 status, or tumor mutation burden. So that shows that it's an entirely different type of an immunotherapy compared to immune checkpoint inhibitors. We've also done intensive screening to find out which neoantigens using whole exome sequencing and RNA sequencing actually are responsible for driving these responses. And what we do is we culture the TIL or the post-TIL apheresis T cells and pulse them with the dendritic cells from the patients with synthesized 11 MERS or 25 MERS, and then screen for using ELISpot for interferon gamma reactivity. And so for the positive hits, what we go ahead and do is we actually do manifest testing to look for the clonal expansion of the specific TCRs, doing sequencing before and afterwards. And that's an expensive way of figuring out which clonotypes actually expand in the presence of that antigen. So this was our results from this interferon ELISA testing, that these infused T cells could recognize a variety of different types of mutations, including some cancer testis antigens that were aberrantly expressed, and including even some fusion events. But I think what the point to take home was that the patients with clinical benefits, and I'm just hiding the complete response patients here, seem to have more neoantigen-specific T cells. And this is consistent with a couple other publications in melanoma. Really not too surprising that, again, if you have TIL, which is confirmed as capable of recognizing cancer mutation, that is going to be likely the more effective one. Here's an example of that. This is a patient, Deborah. She's still in remission to this day. I just saw her last month, and she's doing scuba diving, but she's a former smoker. She had lung adenocarcinoma, low PD-L1, high tumor mutation burden. After tumor harvest, she had nivolumab and had a PR for 10 months, but then had a biopsy-proven new soft tissue metastasis. She had Cy/Flu-TIL IL-2, and then a gradual remission. We gave her consolidation nivolumab for a year after the TIL, and her complete response is ongoing. But her TIL had reactivity to multiple peptides corresponding to unique passenger mutations in her tumor. So out of 56 tested, there was 5 that were reactive, and 3 of them we identified TCRs that were clonally expanded in the presence of those peptides. Almost all of them were CD4, interestingly. And here's another patient, again, predominantly CD4 response, who had a durable complete response, primarily recognizing MAGE antigens. And she is an EGFR exon 19 mutant patient who had progressed on previous osimertinib and other TKIs. So again, this was very interesting that it's recognizing these aberrant MAGE antigens. So this has led us to design another trial, which we're in the process of activating, where we specifically hone in on the oncogene-driven patients. This is a population which is not currently being addressed by a lot of the industry trials. And I think these are a very promising, fit, young, motivated group of patients that are willing to try pretty much anything with a high-risk, high-reward situation and really aim for a home run, if you will, rather than just an infield bunt. And again, our primary goal is to see if this group of patients can overcome the existing, lack of an immune response to immune checkpoint inhibitors, which is classically seen in this population. Usually these patients do not respond to PD-1 inhibitors. So we're also looking into resistance mechanisms. We have funding from the National Research Institute. I'm grateful for this, to use the rich array of samples made possible from the previous trial, including mandatory biopsies at progression. So this is the great thing about a TIL trial, is that you start with so much tumor and you can do a lot with it. Right now we have a hypothesis that A lot of patients are losing persistence of the neoantigen-specific T cells over time, and that has been responsible for their subsequent progression. And we also have strong data that lack of the originally targeted neoantigen has caused subsequent progression. And I'll show you that in a moment. So once again, the presence of TIL-reactive neoantigens was strongly associated with response. As you can see, the orange bars here, the patients that had a long time off of requiring any subsequent systemic therapy, and compared to the blue ones, almost all of them had TIL-reactive neoantigens. And in this example, this patient had a new tumor show up after her treatment a year later, and it contained none of the originally targeted neoantigens. None of the 4 antigens that were originally targeted by the TIL could be detected in the tumor, despite sequencing multiple blocks and despite very high depth of sequencing. So fortunately for this patient, after removing this resistant clone tumor, she remains in remission to this day. So I'm very thankful that the surgery was enough to remove those resistant clones. Now, looking at our entire population, we see that there's a high mutational turnover in the progressive tumors compared to baseline. A lot of new mutations have been acquired, and a lot of the previous mutations have been lost, which suggests that there's this immunogenomic fitness pressure being imposed by the T cells, and that tumors, of course, are able to outsmart our immune system in many subtle ways. In another case, this patient had TCRs that clearly were reactive, strongly reactive to both P53 and KRAS G12V. And I'm listing the TCR beta chains here at the top. What was interesting was that despite having this, and despite, of course, having persistent expression of P53 and KRAS in the progressive tumor, the TCRs themselves were gone from the peripheral blood within months after treatment. And indeed, the patient, despite having an initial partial response, had subsequent progression. And we did an autopsy actually 8 months later, and very few of these TCRs were present in any of the metastatic sites, whether the lymph node, the lung, the liver. So this is really a sign that in some cases, the TCRs the T cells do not persist. It's certainly not in the compartment organs that they're supposed to. And so we've done single cell sequencing, comparing patients with clinical benefit versus no clinical benefit, and the TIL, neoantigen-specific T cells, I should say, within the TIL that are infused do tend to have more of an exhausted programming pathway compared to the ones that had clinical benefit. And just to kind of deconstruct this, the gray dots are the TCR clonotypes, which are expressing which are going primarily in these 2 separate clusters, which are both cytotoxic exhausted clusters, not the type of clusters which traditionally engraft and become memory T cells. So I think that's one of the problems with our infusion process, is that these cells are in culture for 6, 7, 8 weeks of IL-2, and they end up with an exhausted programming pathway, and they cannot persist. So now I'm going to transition just to a couple themes here. One of the themes that we've seen now is that, in the 2nd, 3rd, 4th line setting, anti-PD-1 relapsed refractory stage 4 lung cancer, the response rates aren't quite as durable. That, in fact, although it might be better than docetaxel, we still aren't seeing as durable responses as we like. This was presented at SITC last year. So that being said, a lot of patients are doing excellent with PD-1 naive solid tumors. So whether we're looking at melanoma, head and neck, cervical cancer, or a previous phase 1 trial, the response rates tend to be much better in this situation. Caveat, of course, is that these 3 cohorts had pembrolizumab added. So you almost have to divide this by 2 or maybe subtract 50% off of these numbers, but still it's better than what we usually come to expect with TIL by itself in the PD-1 relapse setting. So we do have a lot of ongoing trials. Instill Bio just opened a trial, Delta 2, which is looking at lung cancer, head and neck cancer, and cervical cancer. And then Iovance is a drug company in the United States that has gone a long way towards commercializing TIL. And there are a lot of other pharma companies that are doing clinical trials as well. Some of these include neoantigen selection methods, where they actually do the hard work of sorting which patients' T cells are actually responding to new antigens, and then enriching for those through a variety of ways. Oftentimes, they're actually co-culturing and stimulating the TIL and actually growing the TIL using those peptides or using those plasmids. But this is very time consuming. It takes, at least 3 months in many cases to get all this process sorted through. And it remains to be seen whether this is really a commercially viable approach. Here's another example. This is Achilles, where they're selecting for clonal neoantigens, which in theory are going to be more important to target. And this requires blood with dendritic cell culture. It requires tumor. It requires sequencing. It requires the lymphodepletion. So certainly these are, the platinum gold Cadillac approach. And the good thing is you're avoiding that stochastic drift. You're actually giving a TIL which has true potency against the cancer cells. But the long turnaround time and logistics, of course, are not trivial. And then another company which has recently actually closed is this peripheral blood stimulation of T cells. And while, again, there's no reason to think these shouldn't work, the approach itself, I think, was taking a lot of time to treat patients. So again, I think this trial is currently closed. So I think the big question we got into there was, well, we're giving this TIL, fine, but how do I know if it's actually targeting the tumors? How do I know if it's actually targeting a tumor antigen? So the advantage of TCRs is that you're not resecting a tumor, and you know exactly what you're targeting because you're transducing those T cells with a vector targeting a tumor antigen, oftentimes an intracellular tumor antigen, something that's expressed by MHC class 1. But one of the disadvantages to this is that it's only targeting one antigen. It's HLA restricted, so it's only applicable to a narrow population, and it depends on HLA expression by the tumor, right? So if the tumor loses class 1 or it doesn't express class 1, this may not even work. It probably won't work. And in fact, there's data from posters like at ASCO this year that showed that it did not work in tumors that do not express HLA class 1. So here's a classic example, NY-ESO in liposarcoma. We see these response rates of around 20, 30, 40%, and they can sometimes be durable. This is something which is very promising. And if this, again, bears forward, this will probably be the first adoptive cell therapy approved in a non-melanoma cancer. Actually, this will probably be the first TCR approved for any solid tumor. Now, traditional TCRs, though, again, we've seen responses in sarcoma, which has high expression of NY-ESO. But realistically, it's really hard to find, HLA class 1 alleles. There's off-tumor on-target side effects, which are still seen with these. And so progress has been very slow, largely related to cytotoxicity, with only a handful of published results, right? So you see a lot of these phase 1 trials never get published because either there's low response rates or because there's a lot of toxicity, or both. So take this with a grain of salt. In contrast, I think a more promising approach is going to be these prefab transgenic TCRs that simply require genetic insertion into a patient's peripheral T cells targeting a public neoantigen. There's much less potential for off-tumor on-target toxicity. And again, as we develop a larger library of these TCRs, this therapy will become more applicable to more patients. Again, the disadvantage is that these are monovalent, right? But the public neoantigens are clonal, like KRAS or EGFR. So it should be present in all the tumor cells right from the beginning, in contrast to some of these, major cancer-testis antigens. So again, I'm really expecting this field will boom because it seems like a great combination between personalization, right? You already know the patient's mutation, and also feasibility and scalability. So some of them use lentiviral vectors, some of them use other sorts of transposases or other technology to insert the TCR. But to me, that's not important. The target is really what's important. At the NCI, if patients don't have the correct mutation HLA pair for a prefab TCR, then there is customized TCR libraries possible where they actually literally create a vector based on that patient's own DNA. So that's a very elaborate approach, and it's great to have a potential product for every patient. And surgery branch also has this bank of prefab TCRs ready to transduce for any patients with these specific P53 mutations. So this encompasses 5% of all solid tumors. Here's an example. This is just a proof of principles showing that this patient had significant relapse in a metastatic breast cancer induced by P53 TCR. So this shows that these libraries truly are scalable and effective. Another great example, just published in New England Journal a couple of weeks ago, was this KRAS G12D TCR validated from a TIL patient and then effective in a previous colorectal patient treated at the National Cancer Institute. And here's the exact same TCR being used at Providence Medical Center in Oregon. And we see a 72% shrinkage in the lung metastases with this pancreatic cancer. So effective across cancers and effective across trials. Unfortunately, this is a really rare HLA type. So again, it's really incumbent on us to seek these TCRs out, grow them, develop them, and test them in phase 1 trials. Unfortunately, this trial using customized TCRs, bag-packed pharma, has subsequently closed, again, because I think of the logistical hampered. So briefly, I'll go over, again, CAR T cells, just for your perspective, as a contrast and compare things. So we've talked about TIL, we've talked about TCRs, and then we've talked about CARs. So CARs have a less risk and expense because we're, again, just harvesting peripheral blood cells, and they also have co-stimulatory domains. So we don't usually use IL-2 with CARs because we believe that these co-stim domains like CD28 or 41BB and other sorts of genetic modifications can avoid the need to do that extra IL-2, which does no doubt add toxicity. However, CARs only target membrane-bound antigens. So the number of targets available for CARs goes from this huge field with TCRs down to a very narrow group, often with CARs. Plus, because you have these activation domains, you have tonic signaling, which may induce some energy or exhaustion in the T cells because they're constantly having that signaling domain on. And above all, the targets in solid tumors are just plain suboptimal. So although there's a lot of CARs popular with startups, as you can see here, historically, there's been very few successes in solid tumors. Everyone wants something to be easy, but unfortunately, I've seen, that these CAR T failures in industry are rarely published, and those suspended or failed first-in-human public trials, they take place, they're abandoned, but meanwhile, new CAR candidates are proposed, often using the exact same targets as the last years or the years before. And this corporate copy-paste strategy from heme cancers into solid tumors overall has yielded unsatisfactory results. So again, the search continues for the optimal targets in CARs or TCRs. So again, off-tumor on-target toxicity is a big consideration, but antigen expression, antigen stability, and density are also, important considerations. One interesting way of compromising or going around this is to use a CAR which actually depends on HLA expression. So with this particular construct, it's only activated in the absence of HLA, which in theory, only the tumor cells will have loss of heterozygosity or loss of HLA class 1. And so one of the requirements to enroll in this trial is you have to have confirmed HLA loss of heterozygosity. And the idea is that you're avoiding the on-target off-tumor side effects that you might get from targeting common antigens like CEA or mesothelin. But you're taking an even bigger haircut in terms of the percentage of patients by requiring this HLA requirement. So this could be a big problem. Maybe homozygous HLA patients, it would not work. So there's still, it's an intriguing concept. So just to go over in conclusions then, we've seen durable responses to TIL in lung cancer, which is exciting. It seems to be agnostic to PD-L1 or tumor mutation burden. I think the recombinant TCRs targeting public neoantigens are probably one of the most promising as well as therapies expand. And while personalized neoantigen selection sounds theoretically attractive, there are a lot of practical considerations. And optimal targets for CAR T cells still are taking a lot of time to identify. So again, I'd like to thank our entire team across the US, including Moffit Cancer Center and our external collaborators for making these studies possible. So I'd like to open up for any questions.
Thank you, Dr. Creelan. That was a very clear overview and amazing results for TIL in metastatic lung cancer. I invite everyone who want to ask a question to open your camera and your microphone and ask the question directly. Perhaps you can raise your hand and I can then ask you to ask your question in order. You can also put it in the chat and I will ask the question for you. I can break the ice, perhaps, Dr. Creelan, with a first question. So you said that TIL may be more efficacious when it's given in PD-1 naive patients or earlier than in the late stage, really immunorefractory setting. How are we going to test this and make this happen for our patients?
Well, so one simple idea, it takes more resources, is to take the tumor when patients are originally diagnosed, or at least before they've received an immune checkpoint, and then have it cryopreserved. Either culture the T cells for several weeks and then cryopreserve them, or we can cryopreserve a tumor digest, a suspension of tumor and T cells, and then culture T cells from that. So that, a cohort of patients treated with those T cells may fare better than a cohort of patients who have tumor resected after a prolonged period of immune checkpoint exposure. However, it's a tough hypothesis to test in melanoma because everyone gets an immune checkpoint really soon. But we may be seeing that, again, as a trend across cohorts, across studies, that there may be somewhat better results in those populations.
Do you think it's going to take randomized controlled trial, or the results will be sufficient in single arm perspective?
Yeah, I think the randomized controlled trials will be required comparing to control for each of those settings. So a randomized controlled trial in lung cancer compared to docetaxel just comparing TIL to docetaxel, you'll have to be using PD-1 previously treated patients. On the other hand, if you have a trial, let's say comparing Keytruda or pembrolizumab versus TIL plus pembrolizumab, that would be another randomized trial in the frontline setting. I think you're not going to have randomized trials comparing TIL to TIL, probably.
Sure. Thanks. We have a question from Alexandre Boutier and then Marie Florescu.
Yes.
Thank you, Dr. Creelan, for the very good presentation. Two questions. The first one is, how do you think AI can help make new drugs that will apply specifically to lung cancer? And the second part, which might fuel data to the AI, is what type of omic data that you think longitudinally can provide better sense if the response is working? I'm thinking specifically on the metabolomics, or something you can get quickly, few hours after the treatment or next day, so you don't have to wait for the overall survival, and then you might have a proxy to that.
Yeah. Okay. So the key question, I think, like you said, is how can AI help us with? And one big thing that AI could help us is with understanding why some patients may respond, predictive markers, not just prognostic, spatial markers. So we know that if a T cell is close to a B cell in an immunohistochemical sample, that those tumors generally tend to respond well to immune checkpoint inhibitors. And that hypothesis may also be true for adoptive cell transfer. So of course, spatial distribution, spatial transcriptomics, I think is something that's still a very developing field. We see new papers coming out in big journals every day on this, but nonetheless, in cell therapy, I think we're still way behind with that. And the same thing true from the metabolic side as well. We are still trying to understand the immunosuppressive tumor microenvironment, which these cells are being supposed to traffic into, and they get blocked. So does that answer your question? I'm just kind of touching on the very simple parts of it.
High enough that I think we're aligned on those two answers. Thank you.
Thanks.
Hello, I have 3 questions. First, thank you for the talk. Very interesting talk. So first question, we have a study at CHUM with TILs, and it's post PD-1 and chemo. So we are taking the biopsy after the patients received their immune checkpoint inhibitors. So this could be done to be. I mean, the patients could have been approached, but the trial is not like that, but could have been approached before and take the biopsy before they're receiving. So what I saw from you is this is what you've done, right? You can figure like something like patients with PD-1 high, and then you take everything, you give the immuno, and when they progress, you do what you prepared everything, and then you do whatever. I mean, the same protocol, and this is what is done. And if I understood well, we're going to have very low responses rate. And my second question is regarding the neoadjuvant effect. Do you think of something in the neoadjuvant setting that could maybe give us some answers? And third question is about, I don't know if you heard about AlloCAR-T. So go ahead.
Yeah, so certainly, I like the idea of banking cells if one has enough resources to do that. With autologous stem cell transplant, we recently successfully treated a patient for whose cells were banked 30 years before they were treated. So if cells can stay in suspension in liquid nitrogen that long, then a couple years should not be a problem. And from a neoadjuvant setting, I think the trouble there, of course, is you never want to impair somebody's ability to get a cure. But it's an interesting area I've never considered to infuse. I think the advantage there obviously is that you can confirm that the T cells have truly trafficked into the tumor. And if you know exactly what you're infusing, you can just do TCR sequencing before and afterwards. But it would be an extraordinary patient that would volunteer for a study like that, I think, because there's a lot of other risks involved. If we had a TCR or a CAR that had almost no side effects and that you could give immediately, then of course it becomes a lot more attractive of a proposition. And I think that leads into the next question, which is allogeneic CARs or other sorts of technology. The problem with allogeneic cells right now, of course, is the lack of persistence, right? That we know the cells get infused and they stick around for like 10 days and then they're gone. And so the trend, my sense is that the trend has been now to actually ramp up the lymphodepletion, to actually increase, at least with CD19 CARs, to increase lymphodepletion and hope that those cells will stick around and persist for longer. It's not an area that I'm close to or as familiar with, but I think obviously off-the-shelf allogeneic cells that you literally can just treat a patient with the same day is extremely attractive. But there's a natural predisposition for our immune system to destroy anything that's foreign. I don't think that there's one thing that's a trend, though, is that this whole fear about graft versus host with allogeneic CARs that have been gene modified to have their CD52 removed and things, that has not been a problem, right? So certainly that issue has been not a problem. It's persistence, it's engraftment. That's been the main problem so far.
Great. We have a question from Habib Mbadassi. Good presentation, please. Can you re-explain slide 13?
Yes. It's a good thing I have them labeled. Okay.
Right.
This slide. Okay, so this is basically showing that patients that have been treated, using TIL with melanoma, and this is the duration of their responses. So the plus means that it's ongoing at the time of the report. Does that clarify it?
If you can open your mic, Habib Mbadassi, you can join the conversation. This is the classical slide that Dr. Steven Rosenberg,
yes,
for those who've been trained with him like me, would show at conference, and that's the way he liked to look at the data. That's great to see this here.
Yeah. Again, these are mostly PD-1 naive patients too.
Yeah.
Yeah.
We have a question from Razvan Diaconescu, an oncologist. Which of the cell technologies would you envision most for the future treatment of lung cancer?
Yeah, so that's a good question. People ask that a lot. I think in the near term, that the TIL is the closest to fruition, that it requires the least, genetic modification. It has the best track record, the most historical experience, and it's probably the closest to commercialization within the next year or 2 or 3 or 4. However, in the long run, one must assume that with all the CRISPR and Sleeping Beauty transposase technology, that we can literally edit the genome of these cells like the same as we can a word processor, that either CARs or TCRs will be a off-the-shelf kind of technology using peripheral blood T cells. Or another potential interesting thing is modifying TIL so that it doesn't require IL-2. Those of us who've been in these trials, and I'm one of those people now, but I remember IL-2 is not easy to give for most patients with lung cancer. So being able to remove that requirement by genetically modifying the T cell to have a coactivation domain or co-stimulation domain really could change things as well for our favor.
Great. Well, there is other people raising their hand or writing in the chat. if I can ask you, so you've mentioned that the lymphodepletion regimen was one of the key key points for this TIL therapy and most cell therapy to be efficacious in patients. You've contributed to a recent paper with Shari Pilon-Thomas about about reactive myelopoiesis, right? The dark side of lymphodepletion. Can you tell us more about this?
Well, I think one of the theoretical effects of lymphodepletion is to kill off some of the suppressive surrounding immune cells, T regulatory cells. Myeloid-derived suppressive cells probably get killed by lymphodepletion, but our postdoc did these suppressor assays where he tested the presence of lymphodepletion actually caused like a lot of cytokines which stimulate myeloid-derived suppressor cells. So in these suppressor assays where he adds back a proportion of cells, you end up seeing much worse T cell responses for these animal models that get lymphodepletion. I think we also took samples from patients and we saw kind of a similar trend where the proportion of MDSCs dramatically increased in the peripheral blood at like the day 7 and the day 14 time points. Of course, some of that could also just be normal reconstitution of the immune system, but those cells were capable of suppressing T cells.
Right, and one thing that was really exciting in this paper is that tocilizumab could perhaps prevent this inhibitory effect from MDSC's reconstitution.
Yes, I'm not as familiar about that part of it. Certainly, we rarely use tocilizumab with TIL, but I think just in about well over half of the patients who are getting CD19 CARs get toci, maybe 80% of them now, right? It's so commonly used. So, and we don't believe that really inhibits the activity of the CAR. So I think it'd be an interesting trial to do to add toci for all patients who are getting TIL and see how those patients respond.
Thanks. We have Antoine Bernard for a question.
Yeah. Hello, very nice presentation.
Thanks.
I'm curious about your single cell data, especially about your observation about stemness.
Yes.
Did you look at it specifically, because this is known after the Rosenberg paper that stemness is very important for cell renewal and persistence, and yeah, did you look at it?
Yes. Well, we did unsupervised clustering to start with of the entire group of TIL cells. So I think we sequenced about 45,000 transcriptomes here, and we did unsupervised clustering. It found about 22 clusters, of which only about 17 were had TCRs mapped to them, because we have VDJ sequencing libraries in here as well. And then we did a few things. We looked at the top expressed genes in the neoantigen-specific T cells. Again, I don't have the data in front of me, but this did not show anything very different from what's already been shown, like CXCL13, so these other kind of classically associated with reactive clonotypes. And then we looked at the ones that had clinical benefit and versus the ones that didn't. And there was some memory cells or some memory markers that were a little bit different, but it wasn't striking. And in fact, some of the resident memory markers were actually worse or actually higher in the no clinical benefit group. And some of the ones that people have been reporting on with immune checkpoint inhibitors like TCF7 and things, those weren't any different. So we're actually planning on publishing on this, and we have like a paper being submitted soon on it. So it goes over the genes a little bit more detail.
Thank you very much.
Thanks a lot. Perhaps we have time for one last question. If there's no one, I have about 20, so I'll pick one. You mentioned one of your great responders. She was on consolidation nivolumab. Did all patients receive anti-PD-1 after their TIL infusion? Yeah.
Yes,
and
for 1 year. So starting at day 30 after their apheresis, after their first CT scan, they resumed the nivolumab. And a lot of people were like, why do that? They already progressed on it. But you've got to think that keeping that going will help prevent those T cells from becoming, going back to sleep again. So I think it was, in hindsight, it was a good idea. Some of the other trials are also doing this too. I think the Instill Bio trial is enrolling PD-1 refractory patients, and it's treating with TIL plus Pembro. So they, I think, have the same theory there. I think there's an Iovance trial, which is, kind of, I think, doing something similar. So there's also this theme, I think, that we took away from ASCO, actually, that PD-1 beyond progression might be a smart thing too. We had this SWOG 1800A trial, which showed that ramucirumab plus Pembro in PD-1 refractory relapsed patients had a pretty substantial response rate. And I think a lot of it is this idea that continuing the PD-1 might be a smart thing to do. If we can. Of course, in the US, we can't get payers to pay for that. I'm sure you're in the same situation. We need randomized evidence.
Right. The universal healthcare system here is bound to other issues and limitations, but we'll find a way, right?
Yeah, I'm same here. No insurance company that reviews a claim would ever approve, and the NCCN guidelines do not recommend it. So yeah. It's still a research question, but I think it would be better than not. Great.
Well, it's already 1 p.m. It was a great pleasure to have you with us today, Dr. Creelan. Thanks for agreeing to present on cell therapy for lung cancer, and I wish you the best. Thanks a lot.
Yes, thanks so much for the chance to present and a really good discussion. Anyone with other questions, feel free to email me.
Thanks. Unfortunately, we cannot hear the applause, but that's the reality of the virtual conference. Thanks. Thank you very much.
Great. Take care. Thanks.